Public health is a multidisciplinary field that works to promote the health of people and communities through monitoring and surveillance, through research, and through intervention practices. Within the scope of public health falls chronic diseases, infectious diseases, as well as injuries and emergencies. Now, there are a few major areas within public health, and these areas include biostatistics, epidemiology, environmental health, health policy and management, and kind of align with this is economics, and the social and behavioral sciences. And social and behavioral sciences includes identifying factors that are going to potentially result in health disparities, as well as promote education. Education is also an important part of epidemiology and environmental health as well. Now, in comparison, One Health is this broader perspective or approach that focuses on relationships among people, animals, plants, and the entire environment. So One Health isn't just human-centric. It's kind of a more holistic or full spectrum perspective approach to health. Basically, humans aren't isolated in their environment, right? They're part of a broader ecosystem. Importantly, we can apply One Health to a variety of different issues, including things like antimicrobial resistance, zoonotic and vector-borne diseases, foodborne illnesses, and even pollution and climate change. The word epidemiology is derived from the Greek epidemios, so epi and demos, which literally means upon the people. It's the study of what is upon the people. So epidemiology looks at distributions, patterns, and determinants of disease, the how and why behind a disease or other health condition in a population, a defined population. Public health relies on epidemiology in a few ways, from surveillance, which is data collection, to assessment, and this is inference based on the available data, and this can also generate hypotheses and be related to hypothesis testing, and thirdly, intervention. So surveillance, assessment, and intervention. Now, there are a few key terms to be familiar with right off the bat here in epidemiology. The first is going to be endemic. So if we're talking about a disease, an endemic disease, a disease that's endemic, this is just the baseline level of that disease in a defined population. And we would compare this to an epidemic or outbreak, which is an often sudden increase in the occurrence of that disease in a population. Now, this may either be kind of a widespread epidemic, or it could just be within a cluster. So a cluster is just a group of localized cases versus it actually being present in epidemic throughout that population. Now, if the spread of disease crosses geographical borders, we would consider this to be a pandemic. So the spread of the disease or health condition across populations, across regions. Other terms, I think, that my brain jumps to when I think about epidemiology are things like morbidity and mortality. So morbidity specifically refers to just having some kind of illness or health condition, and mortality refers to the number of deaths associated with that disease or condition. The occurrence and progression of a disease involve interactions in what we call the epidemiological triad. There's an agent, a host, and the environment. Let's talk about the host first. So a host is an individual that's susceptible to a disease. Now, hosts can have some inherent resistance to a disease or condition, or they may have immunity. So susceptibility almost falls on a bit of a continuum here. Now, the agent is the causative factor of the disease or the condition, and these agents come in a variety of forms. Like they can include physical or chemical stressors, such as high temperatures leading to heat stroke, or maybe radiation, poisoning, dust, fumes, and so on. There are also genetic and psychological agents, inherited traits and other risk factors, social stressors, things of that nature. And there are, of course, biological agents, infectious biological agents, viruses, bacteria, fungi, protozoa, and other parasites. Last in the triad is the environment, and the environment involves the physical abiotic environment as well as the biological landscape and even the socioeconomic or socio-political context that these interactions are occurring within and are dependent on. So the progression of a disease can be divided into two stages. We have the pre-pathogenetic stage and the pathogenetic stage. And pathogenesis is then further subdivided into subclinical and clinical disease stages. Starting out, we have a susceptible host that is then exposed to an agent in the environment. There will be normal adaptation response by the host cells, the host immune system, and if this response is successful, then the disease will not manifest. Everything stops there at that adaptation stage. Otherwise, we move from normal physiological function to pathophysiology, abnormal function. So beginning with subclinical disease, so an asymptomatic incubation or latency period, and then leading to the development of symptoms, which produces, right, this is where we have clinical disease. And eventually a diagnosis as well, hopefully a little bit sooner rather than later. Ultimately, the outcome will be recovery, disability, or death. Both descriptive and hypothesis-based science are used frequently in epidemiology. Descriptive epidemiology deals with the who, when, and where of a health condition, while analytic epidemiology focuses more on the how and the why. So with descriptive epidemiology, we're using a lot of descriptive and observational methods, while the analytic side is more hypothesis-driven, typically involving one or more common study designs, which we'll talk about in a little bit. Now, the purpose of epidemiological studies is to identify statistically significant associations in a target population, which is also called the population at risk. And by statistically significant, we mean that there's a very low probability, less than 0.05 or 5%, that an observed association could have happened by random chance alone. The simplest associations are correlations, which just means that two variables tend to move together. They're not independent. But correlation does not imply causation. I'm going to say that one more time. Correlation does not imply causation. Causal relationships can be very difficult to determine. For simple infectious agents, we can apply Cox postulates, but for more complex conditions, we'll need to consider the Bradford-Hill criteria. I'll explain each of these in a minute. So, causal relationships can be direct or indirect. Infectious agents, they typically act directly to produce a disease, like the parasite Plasmodium. It's the causative agent for malaria. Now, indirect relationships, they're more of like a cascade effect, like how smoking leads to bronchitis, not directly, but by damaging delicate respiratory epithelium and making the epithelium more susceptible to infection, producing bronchitis. So we should also keep in mind that many health conditions, in particular conditions that are not caused by infectious agents, more the chronic and non-infectious conditions, they often have multiple causes, and different causes can have synergistic effects. To establish a causal relationship between an infectious agent and a disease, we can use Koch's postulates. Robert Koch was a German physician. He was also a very early supporter of what's called the germ theory of disease. And the germ theory of disease came out from some work by Louis Pasteur, who, through a series of experiments, demonstrated that it was microorganisms responsible for fermentation and that they don't arise through spontaneous generation in the air or anything like that, that it is only life that leads to life, and that these germs are life. So, with Koch's postulates, it is a set of 4 basic rules or principles that link a specific microorganism to a disease. So we start out looking at a healthy individual and a diseased individual. And the first step is to identify the suspect microorganism in the diseased individual, but note its absence in healthy individuals. Second, we need to isolate and culture this microorganism in the laboratory. We isolate the microorganism from some symptomatic diseased individual. We grow them in laboratory culture. Next, we need to inoculate a healthy individual with the microorganism and see that it produces that disease. Finally, the microorganism needs to be isolated again from that inoculated individual and identified as that same microorganism from the original diseased host. So all of this is very important, kind of a first step in a way, in identifying a causal relationship between microbes in certain diseases and certain diseases. There are a few limitations to Koch's postulates because sometimes healthy organisms can be carriers, asymptomatic carriers of a disease. Other times, some pathogens like certain viruses or prions, they can't be grown in laboratory culture, and that is problematic as well. There's also more complex disease associations out there that we can't really test with Koch's postulates either, like how HPV can actually lead to cervical cancer. So we do need to look at a more complex set of criteria for establishing causal relationships. The Bradford-Hill criteria are a more comprehensive set of things to consider when assessing a potential cause and effect relationship. This isn't a checklist, but it's a very logical framework for organizing and interpreting evidence. There's 9 criteria on this list. The first is strength, and this is essentially the effect size, the magnitude of the effect. If exposure to an agent leads to a major health condition, we're going to be more confident that there's a causal relationship there, compared to if exposure led to just a very minor health issue. Less confident. Second, consistency of data. Are we seeing the same patterns in different samples and in different data sets? Then specificity. Are there other potential factors? Or when we see the effect, is it specifically this suspected causal factor that's involved? Temporality. Now, when we look at the timing, the effect should come after the cause. There might be a time lag depending on the mechanism, but the effect can never happen first. It can only happen second, after the cause, after exposure. Next, a biological gradient. Basically, do higher levels of exposure correlate with higher incidence of the disease or the condition? Specifically, right, this connection. Also, higher exposure may increase severity, which we would call like dose response. Then plausibility. Can we explain the underlying mechanism? Ideally, our explanation is testable and falsifiable. A hypothesis, right? Then coherence. So coherence deals with agreement between the observed epidemiological patterns and laboratory evidence. Of course, the absence of evidence is not evidence of absence, so that's something we might also need to consider. Then experimentation, obviously useful and related to plausibility and coherence. Finally, analogy. With analogy, we might compare the effects of similar factors and the effects associated with them. So, considered all together, these Bradford-Hill criteria can help increase our confidence in a potential cause and effect relationship. A broad range of descriptive, analytic, and experimental studies are used in epidemiology. from individual case reports to ecological and relational studies and up to controlled experiments on animals and humans. We're going to look at three of the most common types of studies in more detail. First, the cross-sectional study. This is a correlational study that simply measures disease and gathers information about exposure factors from individuals simultaneously. So the study, this data collection, it occurs at a single point in time. Case control studies are retrospective, and they study prior exposures of a factor in a group that has the disease in comparison to a control group without the disease. These studies provide stronger evidence of causality, but it's often difficult to identify a suitable comparable control group. Finally, cohort studies are a type of longitudinal study that follows individuals with known exposure over time. They're also a very powerful approach for investigating causality, but a common challenge is retaining participants over time, since these studies can easily last for many years. All right, so looking at these three very common study types in epidemiology, we have cross-sectional, which takes place in the present and establishes correlation. There's case control, which is retrospective, past to present, and it will give us odds ratios. And then there's cohort studies, which are longitudinal, typically from the present to the future, and they provide us with relative risk ratio. There are a variety of sources in general for collecting epidemiological data. The first are vital registries, which include birth and death certificates, as well as related documentation. There's census data, which is very useful in particular for demographic information. There are notifiable disease registries, which are very important public health surveillance tools. They're operating typically at the state or federal level, and they'll monitor the spread of specific diseases, infections, and this can include foodborne illnesses and STDs or STIs. There are legal mandates for healthcare providers to be reporting cases to these registries. Then there are healthcare records themselves. Electronic healthcare records are very powerful tools, and often once patient information is de-identified, they can be used and potentially provided to a variety of epidemiological databases. Population surveys. Population surveys are very important, and they can be used to collect a variety of information about people's demographics, exposure, status, diseases, and so on. Environmental data, by the way, very important as well. Remember, we have the epidemiological triad and the One Health perspective, where it's not just people, there's a lot more context to understanding epidemiology. So environmental data includes information about like contaminants in the air, the water, and so on. Vectors, biological vectors, mechanical vectors. We'll talk more about vectors as we continue. So after collecting data, we need to quantify the disease. And in terms of quantifying disease, we typically don't find a lot of value in reporting absolute numbers. Instead, we consider rates, and a rate describes the frequency of an event in a target population over a period of time. Rates need to include a numerator, a denominator, the target population, and the time period. Also, rates are typically normalized per 100,000 people to be reported per 100,000 people. So, incidence refers to the relative number of new cases in a population over a period of time. Related is mortality, which is simply the incidence of death. And then prevalence or prevalence is the proportion of cases in a population at a point in time or over a period of time. The bathtub model here helps show the relationship between incidence and prevalence, where incidence refers to new cases, and prevalence is the standing amount of cases. So prevalence is equal to incidence times the mean duration of the health condition. Chronic diseases, they can have relatively low incidence but high prevalence compared to acute conditions, which will have low prevalence given the same incidence. Now, when it comes to the epidemiology of infectious diseases, as always, the epidemiological triad is at play: agent, host, environment. But now we only consider biological agents: viruses, bacteria, fungi, protists, and other parasites. These are infectious agents. And now there are additional rates to consider as well. First, infectivity, which is the ability of an agent to infect and multiply or propagate in its host. The lab definition would be the number of infective particles that are needed to establish an infection, but epidemiologically, infectivity is going to refer to the frequency of infection following exposure. So the number of individuals infected over the number that were exposed. Next, pathogenicity. This refers to an agent's ability to actually produce disease. So the number of individuals with clinical disease over the total number that were infected. Last is virulence, and virulence reflects the severity of the disease. So it's often measured in terms of fatality rate, the number of deaths divided by the number of cases. A reservoir is the habitat of an infectious organism or any area it can persist in. And there can be human reservoirs, animal reservoirs, or environmental reservoirs. Environmental reservoirs, for example, are important for tetanus living in the soil and cholera in water. Animal reservoirs are also important in many disease cycles, and an animal-acquired disease is a zoonosis. The infectious organism is transmitted directly from an animal to a human. Also, a reverse zoonosis is when a human transmits a disease to an animal. And humans are common reservoirs for many diseases, and asymptomatic individuals are especially important since they're carriers. They don't exhibit any clinical signs but can be infectious just the same. A very well-known but also very, very sad example was Mary Malone, or Typhoid Mary. She lived in New York, New Jersey-ish in the early 1900s, and she worked as a cook. But she was an asymptomatic carrier of Salmonella typhi, which is the causative agent for typhoid fever. And she ultimately infected around 100 people. She ultimately affected around 100 people, and this led to a handful of deaths. And what I find really sad is that she's forced into quarantine on an island in the East River twice, actually. She was initially released, but then she changed her name to Mary Brown so that she could go back to work in kitchens again, but was ultimately found and then forced into quarantine that second time permanently. And she was there in this Riverside hospital for over 20 years until she died in 1938. Okay, next, moving to transmission. There are two forms, direct and indirect. With direct transmission, we're dealing with direct contact or droplet spread. These are the two major forms of direct transmission. On the indirect side, the first is going to be vehicle-borne, and vehicle is just some kind of typically inanimate object. It is an inanimate object like a fomite. A fomite is just an infected kind of material, like a doorknob or a handkerchief. But vehicles could also be for example, food or water, drinking water. For example, hepatitis A, which is carried in either food or water. Then there are vector-borne transmission, and there are mechanical vectors and biological vectors. Mechanical vectors are sort of like living fomites in a way. Things like flies, and they're just walking around and dirty, and there's like Shigella that's on their bodies. This causes dysentery. In contrast with biological vectors actually harbor and incubate pathogens in their bodily tissues. So, for example, fleas. Fleas host Yersinia pestis in their digestive tract, and this is the causative agent of plague. Yersinia pestis is the bacterium that causes plague. And then finally, there's airborne transmission. With airborne transmission, we're dealing with viral particles typically that are suspended in the air, potentially spores suspended in the air. So not in droplets, just suspended in the air where they can remain for long periods of time. For example, measles. The measles virus can stay suspended in the air. Finally, we have control and prevention. Now, the most important primary prevention is breaking the chain of transmission. This can be done in a few different ways, including controlling animals, controlling biological vectors, controlling environmental factors like air, dust, and dirt, general sanitation, so wastewater treatment, wastewater management, sewage, food, FDA, food safety standards as well. Many different aspects here. Now, another strategy outside of breaking the chain of transmission is to inactivate infectious agents. This can be accomplished in a few different ways, like by pasteurizing dairy products, using UV radiation to sanitize surfaces, using chemical disinfectants, anything of that sort. And then third, we have increased host resistance. So another important form of primary prevention, and with increasing host resistance, we're typically talking about vaccines or just other general health improvements. Things like nutrition and exercise are very important in many different health conditions. Finally, the best advice I'm going to give to you in this whole presentation are the most important personal measures to take. Personal measures for limiting exposure, for breaking the chain of transmission. 1, wash your hands, wear bug spray, use condoms, and cook your food. These are the 4 best personal measures for your protection.